Hello, my name is Rithvik Ganesh, and I'm a student at Plano West Senior High School in Plano, Texas. And this research is titled Efficient Computational Screening of Drug Candidates for the Novel Coronavirus with Applications to Future Disease Outbreaks. The novel coronavirus has caused and is continuing to cause thousands of worldwide deaths along with severe economic damage. The lack of efficient drug candidate screening methodologies has delayed the identification of potential small molecule treatment candidates for the outbreak. This research aimed to address this issue and design a comprehensive drug candidate screening methodology and pipeline utilizing advancements in bioinformatics and artificial intelligence that could be applied easily to the main protease of COVID-19, as well as primary components of future disease outbreaks. In order to be a potential drug candidate truly worth screening in a laboratory, a compound should have two main characteristics. The first is that it should be structurally similar to other semi-effective or partial actives identified in lab-based research, and the second is that it should have a good predicted binding energy and interaction likelihood with the drug target. In this case, the drug target was the COVID-19 main protease. So the first phase of this computational research was to actually screen a data set of over 10 million stock compounds from the Life Chemicals chemical library. And these were specifically screened against three molecules with moderate activity with the main protease: remdesivir, quercetin, and EGCG, a green tea compound. Compounds with high characteristics that were similar to these three known partial actives advanced to the second research phase. In the second phase of research, an algorithm using AI-based neural network architectures was actually implemented to analyze past statistical protein-compound interactions and identify new compounds with key chemical properties. This algorithm was developed by Tsubaki et al. In Japan and was trained using a convolutional neural network and graph neural network, and this was done to analyze key relationships between past actives and inactives. To be more specific amino acid groups on the protein itself, as well as the fingerprints of compounds, form the basis of the interaction probability, which we see in the top and the overall results column of this poster. The final phase of this research involved using a prepared and refined molecular docking trial with binding energy calculations generated by Schrödinger's GLIDE software, as well as AutoDoc-Vina. Of the 10 million original compounds, 5 compounds were identified with substantial binding affinity with the COVID-19 main protease, and they were identified after the culmination of this research with 3 different distinct computational phases. These compounds were then ordered for experimental characterization. And in fact, when these compounds were tested at Dr. Brian Kramer's lab at the University of Washington, one of these 5 compounds was a dose-responsive hit, in inhibiting the main protease as predicted computationally, which is a significant finding. This was a main protease inhibition assay specifically, and the inhibition was measured using fluorescence. In conclusion, this research has led to the identification of an effective inhibitor of the COVID-19 main protease using a newly designed screening methodology. The computational methodology utilized, particularly the strategic use of new algorithms and software, has the potential to be easily applied and scaled for future pandemics, accelerating the identification of life-saving treatments. Thank you very much for listening, and I'll be happy to answer any questions.